All right, we've hit 12:03 and we're starting to level off, so I guess we'll get going. So, hello everybody, and welcome to this week's SBGrid webinar series. Today we have two presenters who are going to talk about some viral related stuff, which is always interesting, and particularly interesting with the current events over the past few months. If you have questions, you can send them to me or one of the other hosts in chat, and we'll moderate those at the end. And so for today, as I said, we have two speakers, both from the Acharya lab at Duke University School of Medicine. So thank you for joining us. Sophie Gobeil and Kartik Mane, and we will start off with Sophie. So with that, feel free to take it away.
Great. Let me share my screen. Okay, so you can see my screen?
Looks good.
Okay, great. So today I'll be talking of the work I've been doing since the start of the pandemic. Actually, I joined Priyamvada's lab right at the start of the pandemic. So we are studying how the SARS-CoV-2 spike D6 protein gene mutation, how a single amino acid change makes a big difference in the structure and protease susceptibility of the spike protein. So I'm happy to say that this work that I'll be talking about today has just been submitted to bioRxiv and is available since last night. So I'm inviting you to actually go see the whole paper. So first, well, I don't think I need really a big introduction for the COVID-19 pandemic. We all know how bad it's been with more than 1 million deaths worldwide, 37 million cases. The United States have been pretty badly affected, but it's all over the world. So that prompted us to really study the proteins and understand how to respond to this global crisis. So beta coronavirus are divided in different clades. There's different beta coronavirus that have been known to infect humans. Actually, there's 7 of them. Of the 7 viruses, there's only 3 that have been responsible for bad epidemics. So the SARS virus, the MERS virus, and the SARS-CoV-2 that we currently are fighting against. And so as you can see, they're divided in different clades, the alpha, beta, and gamma coronavirus. And the SARS, MERS, and SARS-CoV-2 are all beta coronavirus. So why are they called beta coronavirus is due to the fact on their surface, there's the corona, which is the Latin word for crown, which is made by the spike protein that you can see here, which is an homotrimeric glycoprotein at the surface of the virus. And that protein binds to the angiotensin converting enzyme 2 receptors, or ACE2, using the receptor binding domain at the top of the spike. So today I'll drive you through this image, which is, I think, the main figure of the paper. There's 3 different sections of the talk. So the first one, I'll discuss the K986 and V987 proline mutation, which are stabilizing mutations that were introduced in the spike ectodomain construct. I'll then go through the D6 protein G mutation that is been attracting a lot of attention due to the fact that this mutation become highly predominant really fast through the pandemic. And finally, I'll discuss also the fern cleavage site, which is important for the activation of the spike for the infection to the human. So as I said, the first part is going to be on the construct, the ectodomain soluble construct that is currently used, which is called the SGSAS-PP. It's called like this because the fern site is mutated to, which is RRAR in the native protein, is mutated to SGSAS to actually make the construct not cleavable while expressing it in cells. And there's also 2 stabilizing mutations, which are K986 and Val987, change for prolines. This was introduced based on observation of the MERS and SARS spike protein that without those mutations were showing a lot of the fusion conformation. So there was a lot of heterogeneity in the prep of the spike protein. Sorry. And by introducing those 2 prolines, it was shown that the heterogeneity was reduced, and the construct were really the prefusion conformation. So the SARS-CoV-2, those two prolines were introduced in the SARS-CoV-2 construct based on the MERS and SARS. So we first asked, do we really need those two stabilizing mutations, or we could remove them and the SARS-CoV-2 protein would remain still in its prefusion conformation? So throughout the talk, I'll be using three main biophysical methods to measure how the protein is behaving. So I'll use the size exclusion chromatography, which is a way of separating protein based on their mass without denaturating the protein. I'll also be talking of SDS-based gel denaturating type of gels. So basically separating the trimers, the monomers of the trimer. And you can see, again, it separates the protein based on their mass. And finally, I'll be talking also of differential scanning fluorimetry, or DSF, which is a way of measuring the thermal stability of the protein by increasing the temperature. The protein denaturate and expose the tryptophans and aromatic residues, and we're measuring the intrinsic fluorescence of the protein throughout the thermal denaturation of it. So comparing the SGSAS PP versus the SGSAS protein, we can see that they're really similar proteins. They elute on the same, similarly on the size exclusion chromatography. They have a similar pattern of bands on the SDS-based gel, and their thermal denaturation profiles is also really similar. So knowing that the MERS and SARS were showing a lot of differences in their NSEM showing fusion conformation, we then did some negative spin electron microscopy. Here I'm showing the raw micrograph. We did 2D classification, and as you can see here, we're mostly seeing a nice form spike, nice prefusion conformation of this spike, suggesting that the prolines are not necessarily needed in that case for maintaining the SARS-CoV-2 tumor conformation. So we then ask, is the antigenicity affected? So I'll be showing two different types of assays where we add either the spike bound to the surface or the spike flowing over an antibody that is bound to the surface, just to make sure that there's no issues due to the system. So that's basically a way of controlling here. And what we've seen is that both the SGSAS and the SGSAS PP construct really behave similarly in terms of binding to different proteins. So we have ACE2 and CR3022, which requires an RBD up conformation. We have a glycan antibody, which is called 2G12, which binds to an S2 site in a site of the S2 domain in the spike protein. And we have also two convalescent patient antibodies, AB511 and 712, which are binding respectively to the S2 domain of the protein or the S2 site. So the binding site of ACE2 at the top of the spike. So knowing that both proteins are behaving similarly, we ask, is it possible, can we see the structural details? Can we see how the protein is different or exactly similar basically to the SGSAS PP? How does the 2PP affect the structure of the protein? So we obtained the CryoEM structures. So you can see here on the side, the different N-terminal domains, so the NTDs, there's 3 of them, and on the top, the 3 RBD domains, which makes the interaction with the ACE2 receptor. Here is the top view of the spike, and here you can see the RBD going up in the Oneup conformation. So in that data set, we were able to identify both Oneup and 3-down conformation. What we found is that the 3-down conformation, the particles were more predominant compared to the Oneup conformation. And overall, both structures are showing really high, really similar aromatics compared to the structures that were already available in the PDB. So with those structures, we went in and tried to see how this lysine valine mutation to 2 prolines made a difference or not. And what we've seen is that if you mutate lysine 986 to proline, you might in fact affect a potential salt bridge between residue D and impacted with residue D427 because the proline doesn't allow for the salt bridge. So overall, the structures are really similar, but there might be a little bit more interactions in the non-PP version that are possible that we're not seeing in the PP version. So next, as I said earlier, the D614G mutation has been really quickly throughout the pandemic, overwhelming all the sequencing data that was coming from patients. So as you can see, March 1st, we had mostly the D version, which is in yellow. And mid-March, the blue version, which is the D614, already became more prevalent. And by the end of April, it was the main strain isolated. This was identified by Betty Korber and David Montefiore. And so we asked, what does this mutation change in terms of the structure of the protein? So as I said, the D614G mutation is actually making potential hydrogen bonds with interprotomer hydrogen bonds, implicating threonine 859 of the next protomer. And it was also hypothesized to impact glycosylation at a nearby site in 616. So we first produced the protein in the context without the two proteins mutation. So throughout this talk, I'll be using now the SGSAS context. And what we've seen is the protein behaves really well. It's exactly, it's similar to what has been seen without it. So the D and G version are similar in terms of elution profile, migration on SDS-based gel, and thermal stability. We also did a negative stain electron microscopy. And again, we were able to see a nice folded spike, turmeric spike that were in the prefusion conformation. So using this information, we decided to go for a CryoEM structure. Again, we were able to resolve two main populations, the 3-down and 1-up conformation. But looking at these structures, we actually saw that the NTD and RBD were showing a lot of heterogeneity in the maps. So we decided to refine more and more those maps, and we were actually able to identify at least, I'm just showing these, but there's actually more of them, population of subconformation of the RBDs and NTDs moving. And those are all, so here, the one important thing is that both the 1-up and 3-down conformation were populated a little bit like 50%, almost 50% of each, suggesting that the D614G mutation does impact the RBD NTD dynamics or the conformational exchange going on in the spike protein. So we next use the 1-up conformation and decided to go see through the details of the structure that we could capture. Here, I'm going to use this color scheme where the RBD is going to be in red, the NTD is going to be in green, the SD1 domain is going to be blue, and SD2 domain, which contains both the furan site and the D614G mutation, is going to be in orange. There's also a connector that goes from the NTD up to the RBD up to the NTD, which we call the N2R for NTD to RBD domain. And that connector, which is 28 amino acid long, is lying between the NTD and the SD2 domain. So aligning all the 1-up protomers, all the protomers for the 1-up structures, you can see in dark red, here is the up RBD domain, while in salmon or lighter pink, it's the down conformation. We could see that the RBD goes a really big change of more than 40 angstrom. And the NTDs also show some variability between the 3 different protomers. Interestingly, we captured that between the SD1 domain, between the 1-up and the 3-down conformation, the connector, the section of the connector N2R was also showing a lot of the variability in the orientation it was adapting, and it was moving by more than by approximately 7 angstrom away. Interestingly, the SD2 domain, which I said earlier, do have the G614G mutation and the furan site, really didn't show much variation. We had approximately 2 angstrom difference in the biggest, the most differing section of the SD2 domain. So we then decided, because this structural analysis depends on how you align the protein. So we decided to use the DDM analysis, which is a different distance matrices, and it's a superposition-free comparison of 2 different structures. So we compared all the different protomers from the 1-up structures. And what we found is that the NTD does respond to a change sorry, the SD1 domain respond to change in the entity, as you can see here by the blue, and the N2R, the linker, does respond to entity change and changes both in the SD1 and SD2. So even though this is just a beta strand that goes from one domain to another one, it does respond to the environment that is changing due to the SD1 and SD2 conformation. The other really interesting phenomenon that we observe is that somehow the SD2 domain is really making an anchoring point for blocking all the differences that we can see in the NTD and RBD domains, and not translate the motions to the S2 domains. So it's a stable anchoring point in the spike protein between the RTD and the NTD. So the next question that we ask is, knowing that the D614G mutation is in the SD2 domain, and that this domain also has the fern site, which is essential for the spike to be cleaved and then do the infection, we ask if it was the D614G had any impact on the fern site. So we used the SGSAS context, and we decided to introduce a new cleavage site right where the fern site would usually be, which makes the construct now susceptible to HRV3C. We did that because natively our expression system, which is 292F cells, do express furin. So that means that while expressing the protein would be cleaved. So by having an exogenous site, which is HRV3C, the protein would remain as a spike throughout the expression. So as I said, we have the HRV3 site exogenous. We purify the protein using our usual method, and then we add the enzyme outside of the cells, so doing a digestion in vitro. And then on gel, we would expect that without the HRV3 enzyme, you would have one band, but during digestion, you would start to see the S1 and S2 domains, which are one side or the other side of the protein. So we first purify the protein, seeing nicely folded spike, showing one main band on the gel, and then do negative stain electron microscopy, showing nicely folded spike. That's exactly what we expected. And then we added the HRV3C enzyme, and this is where we saw a really interesting phenomenon. So the D version, so the SHRV3C, was less susceptible to digestion compared to the D614G mutation. So by having this mutation, somehow the fern site is more exposed and more accessible to HRV3C. And you can see that not only how much protein is digested, but also the speed at which the digestion is going is completely different. So within 10 minutes, the whole HRV3C D614G protein was almost digested, while the D version was not reaching full digestion even after 24 hours and adding more enzyme. So we then decided to try the restored furin site. So it's exactly the same construct with and without the D614G mutation, but we restored here the furin site to RRAR. So that means that during expression, because furin is endogenous to the system, we would have both the full spike and also some cleaved spike. So we would use our usual strategy for purifying and probably only capturing the fully folded spike, or maybe some part of the also digestion portion of it. And then on gel, we would see the different bands corresponding to the full S protein and the S1, S2 domains. So we did purify the proteins. As you can see on size exclusion, we have probably agglomeration of the protein, some sticking going on, but we also had a peak of nicely folded spike. On gel, we can see the S protein that is making the full protomer, but also the S1 and S2 domain that were cleaved during expression. What's interesting here is that even though we kind of have different ways of excluding smaller molecular weight size proteins, we still have the S1, S2 domains that are trailing during the purification. And as you can see here on the NSCM, we were able to still capture nicely folded thymeric spike after the purification. So we then added the furin enzyme to our size exclusion purified protein. And as you can see again, exactly as what we've seen, with the HRV3 engineered system, the fern, the D version is not cleaved, while the D614G version was more cleaved. So we decided to take that portion of protein that was cleaved and do size exclusion to try to eliminate the band, to purify it. And we were able to capture a thermeric form that was eluting as a thermeric form. And then on gel, as you can see here, we had a fully cleaved S1, S2 spike, but that was still eluting as a fully thermeric spike. So that suggested that the spike was still opening up together, even though it was fully cleaved by fern. So we did a negative stain electron microscopy. And again, that confirmed that the spike, even if it was cleaved, it was still loading up as a thermeric construct. So we decided to obtain CryoEM structures of this. And what we found is that the 3-down conformation was now becoming more predominant compared to the 1-up conformation. That suggests that the fern cleaved spike has a different dynamics on the RBD NTD domains compared to the non-cleaved version. And we also looked in the structures and we saw that there's a site in the SD2 domain that does respond to the NTD RBD motions, which is not too far away from the fern cleavage site. So this was also captured by the antigenicity assays. So as you can see, if the fern cleaved spike, which has more RBD downs, there's a decrease in binding for the antibodies and S2 that targets that needs an RBD up conformation. Yeah, so with the red line here, which we see a decrease. So overall, I think throughout this whole presentation, well, the first main message is that both the SGSAS PP and non-PP versions are similar in terms of their structure, biophysics, and antigenicity. The D615G mutations alters the dynamics of the RBD domains, so how the 1-up to 3-down ratio, and it also enhances the furin susceptibility of the spike protein. Furin cleavage does also further alters the dynamic of the RBD domains, as we saw in the change in antigenicity. And overall, I think here the main message is that this SD2 domain is really an anchor that blocks the motions from the NTD to RBD and not translate to the S2 domains. And the linker that connects the NTD to the RBD domains, which is called the N2R, is a modulator of the change between the RBD and then the NTD. So it's really a vector of translating the information between the 1-up conformation and where the NTD has to go. So this is just a little snapshot of what we saw in terms of how many 1-up versus 3-down, how many particles of 1-up, 3-downs, the antigenicity change, as I said, the D614G that's completely changed, and the protease susceptibility. So all of this is important for vaccine design because it helps us understand how the dynamics might affect, how the dynamics of the protein might affect binding of different antibodies. So I want to take time also to acknowledge everybody in the lab who's worked on this with me, Katarzyna and Shayna, Priyamvada. That was really an incredible effort that we had to do with this. We learned through on the fly. I want to also acknowledge the AIMS group, which was doing a lot of ELISA assays for us. Kevin Saunders, Gregory Stampowski is also producing some antibodies. I want to acknowledge the Smith for allowing us access of microscope time, the NIH for the money, computer, the Duke University Computer Center for giving us access to the computing office, and then the NCCAT for access to the microscope as well. And I want to again say, well, the paper is now on bioRxiv, so I invite you to go read this. And I'll be happy to take any questions.
Great, thank you, Sophie. That was a really interesting talk, and we do have questions for you. So to start off, could you comment on how extensive the spike glycosylation impacts the different mutations, and if that has any impact on the biophysical measurements?
Yeah, so glycosylation, yeah, for sure. It could hide completely sites. So we do produce in human cells just for that reason. We want to be sure that we're as close as possible to the natural system. What we've seen, the D versus G difference in protease susceptibility is probably linked to partly exposing more the site, but also having some glycans maybe moving away. I don't know. That would be a really interesting question.
Thank you. I guess related to your earlier, the K986P and V987P, you were conjecturing that those, or your model for those was that they might be involved in a salt bridge, and that would be responsible for the changes that you were seeing. Do you know if there are any other mutations to those sites that are not proline that are also similar, or is that something that hasn't been investigated yet?
Yeah, so those two mutations are actually non-natural mutation. They're stabilizing mutation. They come from different other systems. So for example, in the HIV CYP proteins, there's also two mutations that have been prolines helping stabilizing the protein conformation. And it's also been seen for SARS and MERS. So it's really engineered stabilizing mutations. Usually, I don't think I've seen any reports on residue 986 and 987 being mutated to other positions, other amino acids. But one thing we've been looking into is how does it compare to MERS and SARS? Because there's some structures also where we can see those same amino acids, so the K and V. And for MERS, it's actually not in K and V, it's a valine and I think an isoleucine, so it doesn't allow for a salt bridge. And for SARS, what's interesting is that the D1427, which would be implicated in that salt bridge, lies further away. So that suggests that the salt bridge might be more transient and might not have necessarily the same impact for SARS-CoV-2.
Thank you. And I think last kind of combination of two questions that are both related. So for the conformational flexibility, when you were measuring the rates there, are all of those measurements from the CryoEM populations? And are those things that have been investigated or collaborated by molecular dynamics simulations?
Not on those constructs yet, but that's something we're actively thinking of going through. We actually published, so Rory Anderson published a paper with us and Priyamvada on reporting on the method where we a vector analysis between the how does the NTD and RBD motions do correlate. And so, yes, this is something we're thinking of doing now, using all those different structures, especially the set of 8 that I'm showing for the D614G. Clearly, there's a lot of information that we can extract from this.
Great, thank you. So with that, actually, Wolfram, you have a question, so would you like to unmute yourself and ask?
Yes, do you have a sense if or how these multitude of structural studies on the spike protein, for example, might be influencing the early generation vaccines? Or do you think that the vaccine developers doing their own unpublished structural research like yours?
I don't. So what we've seen is antigenicity and every measure, biophysical measure that we have are similar. So I don't really think that it's going to change much of the vaccine development. But I think the RBD to NTD dynamics that we're seeing, especially in the D614G, is going to really impact, has some changes in the way we approach vaccine development. So I don't think the two prolines are the case, but I think the D614G and the mutations might be more. And there was actually last night a paper that came out on Biochemical as well, showing that if you bind the ACE2 to the RBD, it increased heterogeneity in the S1 domain, and it completely, it changed the S1 domain basically. And so the idea is if you have also the D614G at the same time, maybe that just contributes to a better shedding of the virus and then more an increase of infectivity that was already reported. So yeah, that's an interesting observation that came up really well with what we've seen.
Thank you.
Great, thanks. And that looks to be all of our questions. So thank you again for presenting.